Okay, we're rolling. And I had raised the question last Wednesday of what exactly are these genes? So we had started with the question as to what are these crazy little things called genes? Gregor Mendel has worked out that they must exist by statistical study of the results of his crossbreeding experiments, but what exactly are they? To get there, we have to kind of go back, and I reviewed the discovery that life is made up of cells, and when eukaryotic cells divide, as seen under the microscope, the nucleus appears to turn into a set of thread-like objects, and these, of course, are called chromosomes. Chromosome meaning colored body, because chromosomes stain very strongly with various artificial stains. This is not it, but they stain very strongly with methylene blue, which is a stain that I know you've used. And when the cell divides, they go through a little dance called mitosis. And we went over the stages of mitosis. When a cell is not in cell division, we say it's in interphase. And then in prophase, the chromosomes appear. In metaphase, they line up in the middle on the equator of the cell. In anaphase, they separate and move to opposite ends of the cell. And in telophase, they form new nuclei. And usually, although not always, the rest of the cell divides when the nucleus is reaching telophase. And that division of the whole cell is called cytokinesis. So that's where we were. And I'd mentioned that very careful microscopic studies showed that at the start of cell division, each chromosome is made of two parallel strands. There's one, and there's one, which is cool. I can go back and forth with these little arrow keys and make it computer animation. This is practically Pixar. And those two strands are called chromatids, and they're joined at a point in the center, right there, called the centromere. And here in this other chromosome, you can see this is one chromatid. There's the other chromatid. And they're joined together at the central spot that's called the centromere. So that's just a recap of what we covered yesterday. You certainly are responsible for knowing all of the, come on, Wagner, knowing all of the stages of mitosis, the order that they come in, and the major events that happen in them. So there's the chromatids. At the start of meiosis, each chromosome in a cell consists of two chromatids, two strands joined at the centromere. At the end, each chromosome consists of only one chromatid. Remember, the chromosomes appear, they line up, and then at anaphase, the centromere breaks, and each chromatid goes in opposite directions to opposite sides of the cell. So at the end of cell division, each chromosome consists of one chromatid, but you can watch mitosis happen if you're patient. It might take 30 minutes to an hour. It does, with patience, you can watch it. And if you watch it happen and then let that cell just keep living, and it'll stay in interphase for a while, and it will just metabolize and grow and do whatever it does. And then when it divides, when the daughter cells divide again, each daughter cell starts with two chromatids per chromosome. So when we can't see the chromosomes anymore, they consist of one chromatid. When they reappear, they consist of two chromatids. So somewhere during interphase, each chromosome has to make a new chromatid. The chromosomes are always there. You can't see them when the cell is in interphase. You can't make them out as separate little entities. Why that's the case, we will have to save for a little bit later. I promise I will get to that. But just know that when a cell is not dividing, the chromosomes are still present, but you can't make them out as separate entities under a light microscope. And somewhere during interphase, each chromosome must somehow sprout a new chromatid. And so we now divide a cell's life into what we call the cell cycle. Down here, the part that's labeled M, I've called, that's mitosis. That's prophase, metaphase, anaphase, telophase. And at the end of mitosis, every chromosome consists of a single chromatid, kind of like that. Then the cell enters what we call G1. G just stands for growth. The cell just grows and metabolizes and, does whatever it does, calls on its cell phone, and that was a joke. So there's this time period where the cell just hangs out. That's followed by the S phase, a time in which the cell somehow, we can't see what it's doing yet. We will, but we can't see it yet. The cell must somehow magically create new chromatids. And then after the S phase comes a stage called G2, where the cell will grow some more and prepare to go into mitosis. And right when mitosis begins, we can finally see the chromosomes when the cell goes into prophase, and each chromosome consists of two chromatids. So somewhere in the S phase, each chromosome sprouted a new chromatid by a process that we cannot yet see. And we will get to what happens in more detail, but leave it for right now. So we know that some mystery happens in the S phase, where each chromosome gets a new chromatid. By the way, the actual timing on this depends on what cell you've got. There are some cells where there's virtually no G2, where as soon as S is done, the cell goes right into mitosis, and there's practically no G2 worth talking about. There's others, like many, although not all, of the nerve cells in your brain go into G1 and they stop. They'll never divide again, and they're just permanently stuck in G1. There's others where S might happen pretty soon, and then G2, they might stay in G2 for a long time. It depends on the cell. Different cells, different organisms have different timings of the cell cycle. Okay, why should any rational person care? Well, mitosis is the way that single-celled eukaryotes reproduce. It's how you get one amoeba to become two. It's the way that multicellular eukaryotes develop. And when we talk about development in biology, we mean the process by which you go from a single cell to 2, 4, 8, 16, 17 trillion. 37 trillion in you. All of your cells, with a couple of key exceptions, were produced by mitosis, and they all ultimately go back to the single fertilized egg that started out you, that started the development of your body. All of those cells, except for a couple, came about through mitosis. It's still going on in your body because every day you lose 50 to 70 billion cells, sloughing off of your skin cells, sloughing off of other cells that break down very quickly, like the cells that make up the cornea of your eye That's the part you put the contact lens on, or especially the cells that line your intestines. They're always breaking down and dying and getting sloughed off, and they have to be replaced by new cells. So every day, your cells undergo mitosis something like between 50 and 70 billion times just to maintain you as you are. Mitosis is one of the ways in which you can repair wounds. You can grow new tissue to replace wounds that you've lost. You're not nearly as good at regeneration as some animals, but organs like your liver, if you lose half of your liver, it's actually very good at growing back, and that's due to mitosis. And then ultimately, cancer is mitosis with a stuck accelerator and broken brakes. Cancer is what you get when a cell in your body begins dividing and can't stop. We're probably not going to have time to go into exactly how they do this. It's a pretty big field, but the cells in your body have ways of regulating the cell cycle so that they don't, so they divide only when they're needed. But those mechanisms can fail. And when that happens, you can get a cell that starts dividing very rapidly and cannot stop dividing, and gets big enough that it starts seriously draining your body's resources. That's basically cancer. And a lump of cancerous cells that are all dividing very fast is, of course, a tumor. So mitosis happens all the time. You encounter it in lots of different contexts. But if you thought that was bad, we actually have to go through a second type of cell division. Dun, dun, dun. And this one is trickier. And it is, in fact, called dun, 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 meiosis. M-E-I-O-S-I-S, meiosis. Now, a little bit of context. In you and in animals in general, meiosis is the way that you get eggs or sperm, or in some animals, both at the same time, or in other animals, both at different times in the life cycle. Long story there. With plants and protists, it can get a little weird. Well, not weird, but it can follow different patterns that we just don't have time to get into. But at least as far as animal biology goes, meiosis is the way that you produce eggs or sperm. And by the way, this word will pop up again, but an egg or sperm is called a gamete. A specialized cell that is used in sexual reproduction is a gamete, whether it's an egg or a sperm, or whether it's just a reproductive cell that is neither, which is what you might get in some seaweeds, for example. Anyway, to understand mitosis, I've got to give you a little more background on chromosomes. Chromosomes in one cell are not all identical. It's possible, and in fact, medical technologists do this regularly. We can take some of your cells, photograph them as they're dividing, and sort out the images of the chromosomes and count how many the cell has. And it turns out that some chromosomes are long and some are short. Some have the centromere that's close to the actual center of the chromosome. That's called being centric, by the way. And some chromosomes have the centromere a little off center, closer to one end than the other. And we call those acentric chromosomes. And then finally, depending on the staining technique that you use, you can often make out that chromosomes are banded. They have striped patterns on them. I'll show you what those look like in a bit. And it turns out if you do this, if you photograph all the chromosomes in a cell and you start sorting through the photos, it turns out that in a typical eukaryotic cell, there are 2 of each chromosome type. And a cell that's got 2 of each chromosome type, we say is diploid, from the Greek word for double, diplos. So a cell with 2 of each chromosome type is diploid. Almost all of the cells that make up in your body are diploid. So these happen to be chromosomes from a normal human female, and there's 46 of them. That is the pattern for normal human beings in general. We've all got 46 chromosomes per cell. And not only do we have 46, but we can sort through them by size and centromere position and banding pattern. These have been stained with some kind of fluorescent stain that stains parts of the chromosomes red and parts of them yellow. And so you can see up at the top left, we have 2 copies of a long chromosome that is centric with the centromere right there in the middle, and banding patterns that include a big yellow strip on the tips. And we call those chromosome 1. Chromosome 1 just because it's the largest. Chromosome 2 is only slightly smaller than chromosome 1, and you can see it's got a different banding pattern. And we have 2 of chromosome 2, 2 of chromosome 3, 2 of chromosome 4, and so on. We just sort these down to size, all the way down here to chromosome 21, chromosome 22, and then there's a pair of chromosomes right here called X chromosomes, which I will talk about later. Just know for right now that there are 23 pairs of chromosomes in a typical human cell. And we can pair them up by size, by centromere location, and by banding pattern. You can see that the members of a pair of chromosomes have pretty much the same pattern of red and yellow stripes or bands on them. Different animal and plant species have different chromosome numbers. These happen to be mice chromosomes, and mice have 40 chromosomes per cell, arranged in 20 pairs. Horses have, I believe it's 64 chromosomes in 32 pairs. Fruit flies have 8 chromosomes arranged in 4 pairs. A very small plant called the adder's tongue fern happens to contain 1,260 chromosomes per cell, which you can arrange in 630 pairs, and so on. Closely related organisms usually, although not always, have similar chromosome numbers. You've got 46. Chimps and gorillas have 48, for example. But there's no real relationship between an organism's complexity and how many chromosomes it's got. It's just a number. And you don't need to memorize any of those numbers. I don't even particularly care if you remember that humans have 46 chromosomes. Just know that chromosomes come in pairs like this. Okay, many eukaryotes form specialized cells for sexual reproduction called gametes. And that's a word I used earlier. Usually gametes come in 2 forms, big and small. Large gametes are called eggs. Small gametes are called sperm. Sperm do not always swim. Eggs are not necessarily really big. Human egg cells are, I forget, but it's barely naked eye size. You could see a human egg cell, but it would just look like a really tiny dot. Human sperm cells are maybe 100 times smaller than that. In many protists and fungi, the gametes are all the same size, so we don't divide those up into eggs and sperm. Sometimes we can divide them up into separate mating types, and we might call these plus type and minus type, or type A and type alpha. Doesn't really matter for our purposes here. Sometimes gametes come in two different forms, and sometimes they don't. And here's the thing, gametes only have one of each pair of chromosomes, and we call this being haploid. So when you started out your existence inside your mom's fallopian tubes, your mom produced an egg cell that had 23 chromosomes, one of each pair of chromosomes that she has. Your dad produced sperm cells with 23 chromosomes each, one of each pair of chromosomes that he had. And when Mrs. Egg and Mr. Sperm got together, and Mr. Sperm managed to break through the membrane and enter Mrs. Egg, their nuclei combined, and you ended up with a fertilized egg cell that contained 46 chromosomes, 23 of which came from mom and 23 of which came from dad. And when a cell has only one of each pair of chromosomes, we call it being haploid. All right, so yeah, here's a human egg cell being swarmed by a bunch of human sperm cells. They're all trying to penetrate into the egg cell, and normally only one of them will make it. The egg cells actually got ways to block the entrance of all of those sperm cells, except for the first. Both the egg cell and the sperm cell have one of each pair. So one of each of 23 pairs of chromosomes. So the egg and sperm both contain 23 chromosomes each. Here's an egg and a sperm that have fused together, and their nuclei down there are in the process of coming together. And when an egg and a sperm fuse, the resulting cell is called a zygote. And this zygote now has two of each chromosome. This zygote now has 46 chromosomes in 23 pairs. One of each pair came from mom. One of each pair came from dad. And incidentally, meiosis is how you go from a diploid cell with two of each chromosome to make haploid cells that have one of each chromosome. Meiosis is sometimes called reduction division for that reason, because you go from a typical cell that's got two of each to eggs or sperm that contain one of each. An error in meiosis can cause an egg or sperm to accidentally have two copies of one chromosome or possibly be missing a chromosome. These are some human chromosomes again. They come in 23 pairs labeled 1 through 22, and then a pair of X chromosomes that I'll talk about later. But right there, whoops. Okay, that just flashed once. So right there, you'll notice that the cell that these chromosomes were taken from has 3 copies of chromosome 21. Not 2. So one of the parents, it could be either the mother or the father, it doesn't really matter which, because of a mistake in meiosis, which does sometimes happen, one of the parents contributed not one of each chromosome, but one of almost all the chromosomes, but 2 copies of chromosome 21. By the way, people born with 3 copies of chromosome 21 are born with what's called Down syndrome. Generally developmental delays and some physical features that tend to be typical, like an enlarged tongue, sometimes heart defects. Well, it's Down syndrome. Most of the time, if you get an extra chromosome, a baby with an extra of a chromosome that's other than 21 usually just miscarries and is never born at all. You can have babies that are born with 3 copies of chromosome 13, that's called Patau syndrome, or 3 copies of 18, that's called Edwards syndrome. They are usually pretty severely physically disabled. A lot of things go wrong, and they usually don't live very long. There's a few cases of them surviving into childhood, but it doesn't happen very often. Okay, checking the chat here. What about intersex people? Can I get to that later? You can also get extra copies or missing copies of these chromosomes down here called the sex chromosomes. Let me leave that until I can cover sex chromosomes more in the detail they deserve. You can get people with extra copies of a sex chromosome or missing copies, and this can have interesting effects. But let's not jump the gun. Okay, so how does this meiosis work? It's a double division. In making sperm, you start with a diploid cell, and guys, you've got a bunch of these in your testes. They're called spermatogonia, and they're diploid, and each spermatogonium cell divides and then divides again. And we call this meiosis 1 and meiosis 2, and you end up with 4 cells at the end. So every spermatogonium divides to produce 4 protosperm that mature into sperm cells or spermatozoa. In women, well, in female animals in general, it's a little bit different. There's a wrinkle to it that I'm not sure I'm going to have time for. There are 2 divisions, but you have cells in your ovaries called oogonia. And those also divide twice, but you end up not with 4 egg cells. You end up with 1 egg cell and a few throwaway little rudimentary cells called polar bodies. I'm not going to be able to get into that just for reasons of time. No, the process looks a little bit different, but still, meiosis is how you make egg cells from these progenitor cells in your ovaries called oogonia. So before meiosis, a cell goes through G1, S, and G2. And when meiosis starts, the cell has 2 of each chromosome, and each chromosome has 2 chromatids. Meiosis 1 of each pair of chromosomes goes to each daughter cell. So at the end of meiosis 1, you have 2 cells with 1 of each chromosome. And then in meiosis 2, each chromosome splits and 1 chromatid goes to each daughter cell. Meiosis 2 looks a lot like mitosis does. And each one of those divisions, meiosis 1 and meiosis 2, can be divided into prophase, metaphase, anaphase, and telophase. So we can have prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, and telophase 2. I'm not making you learn all of those, but here's the basic accounting that you need to be able to keep track of. Before meiosis starts, we start with 1 cell. It's got 2 of each chromosome. And each chromosome consists of 2 chromatids. I'm only drawing one pair, but of course in cells there would be more than one pair, but I'm just drawing one at a time. At the end of meiosis 1, you have 2 cells, and each one contains 1 of each chromosome, and each chromosome consists of 2 chromatids. And then when we're done with meiosis 2, you have, in the case of sperm, you've got 4 cells. And these cells, by the way, will sprout a tail and get rid of most of their cytoplasm and end up becoming sperm cells. You end up with 4 cells. Each one contains 1 of each chromosome, and each chromosome is made up of just 1 chromatid. And again, egg cells have a slight wrinkle because when egg cells are forming through meiosis, the cell division is not equal. So you end up, in the case of making eggs, with 1 big cell with 1 of each chromosome, and then 3 little throwaway bits called polar bodies that are pretty much discarded. Again, I'm not going to go into that very much, but know that it's not quite the same in eggs versus sperm. So meiosis 1 starts out looking like mitosis. These, by the way, are pictures of cells undergoing meiosis in the anther of a lily flower. These are going to become parts of pollen grains. Pollen is not exactly plant sperm, but it would be more accurate to say that it does contain plant sperm. And meiosis is what it takes place when plants are making pollen. The plants around here are getting ready to undergo an awful lot of meiosis and spread it all over your cars in the form of yellow dust and an awful lot of sneezing, right? That's what pollen does. So meiosis 1 starts out like mitosis. In prophase 1, the chromosomes condense. On the left, you've got early prophase, and then on the right, it's later, and you can see the chromosomes get thicker and clearer. And I promise we will get to later exactly what is going on here, what's making them get thicker. But in meiosis 1, something else happened. When prophase 1 hits, each chromosome pairs up with the other member of its pair. So we didn't see this in mitosis. But at the beginning of meiosis 1, each chromosome 1 will pair up with the other chromosome 1. Each chromosome 2 will pair up with the other chromosome 2, and so on. And what you're looking at is a bunch of chromosomes that have paired up. This thing that looks like an X in the middle, that's a pair of chromosomes. That's a pair of chromosomes. That's a pair of chromosomes. This thing that looks like a fish, that's a pair of chromosomes.
Can I ask a question?
Shoot.
So like you said, if there are 3, would the 3rd one just be left out, or would it still pair up with the other ones?
Okay. In the case of somebody that's got 3 of one of the chromosomes, if you've got an extra of 1, let me see. In some cases, the 3rd one is just left out, and meiosis happens, and the 3rd one doesn't pair up with anybody. In other cases, you can end up with meiosis being so disrupted that the critter can't make eggs or sperm at all. And again, it's a situational thing. It's going to be different for different species. Okay. When they've paired up, the paired chromosomes undergo something called crossing over. And that means that they break and swap equivalent pieces of each chromatid. So this right here is one chromosome. And what I'm outlining right here is one of its chromatids. And there is its other chromatid. I think. And there's the other chromosome. And I'm outlining its chromatids. And you can see they're kind of woven into each other. And yeah, what is going on here is that the chromosomes have not only paired up, but they are exchanging equivalent pieces with each other. It would be kind of like if I could stand next to you and my arm and your arm suddenly switch places. So I stood next to you and suddenly your arm is on my body and my arm is on yours. That's kind of weird when you think about it in those terms, but chromosomes do it all the time. They'll pair up and swap equivalent pieces of each chromatid. That's called crossing over. That is, I will try to get to a little bit later as to why that's important. But for right now, just know that that is one thing that happens in meiosis, but not mitosis, this pairing up and this crossing over. And meiosis 1 ends with the formation of 2 haploid cells. So this would be anaphase 1, and one of each chromosome pair moves to each daughter cell. Actually, if that's beginning to divide, that's more like telophase 1. And then meiosis 2 is exactly like a repeat of mitosis. The chromosomes appear again, they line up in the middle, they separate at the centromeres, and each chromatid moves to opposite ends of the cell. So this looks like anaphase 2 as the chromatids are moving to opposite ends of the cell. And there's our end result. There we have telophase 2 with 4 haploid cells. Each one of these has one of each pair of chromosomes, and each chromosome consists of only one chromatid. Ta-da! Okay, who the hell cares? You may be asking yourself. Here's why this matters. Here's why people started getting excited about Mendel's rules. Mendel had worked out that genes come in pairs. An organism inherits one of each pair of genes from each parent, and then it passes on one of each pair of its genes to each one of its offspring. Here's the weird thing. If you follow chromosomes through meiosis, the same thing happens. You receive one of each pair of your chromosomes from each parent, and you pass one of each chromosomes to each of your offspring. So you have 2 copies of chromosome number 1 in every one of your cells, except your eggs or sperm. And you got 1 copy of those from mom and 1 copy of chromosome 1 from dad. And it's the same with all the others. You've got 2 copies of chromosome 2 in almost all of your cells. One of those copies came from your mom, and one of those copies came from your dad. And if you decide to have kids, you will pass on one of each of your chromosome pairs to each one of your offspring. My son has 2 of each chromosome in almost all of his cells, and he got one of those of each pair from me and one from my wife. And so here's the point. We've been working on opposite sides of the jigsaw puzzle, but hopefully the pieces it's starting to become clear how those pieces fit. Mendel works out that genes come in pairs and you inherit one of each pair from each parent. Microscopists in the 1870s worked out that chromosomes come in pairs. You inherit one of each pair from each parent, and you pass one of each pair to each of your offspring. That's exactly what Mendel worked out for genes. And this must be really important, because if meiosis goes wrong and you have an egg or sperm that ends up with a missing or an extra chromosome, the zygote that results either won't develop or it won't develop properly. So genes and chromosomes are following the same rules. So what's the link between them? Well, we know that genes and chromosomes aren't exactly the same thing, for the simple reason that there are a lot more genes in an organism than there are chromosomes. Peas have 14 chromosomes in 7 pairs, but they've got a lot more traits than 7. They've got a lot more traits that follow Mendel's rules than just 7. So clearly a chromosome is not the same thing as a gene, because you've got many more genes than you have chromosomes. You have 46 chromosomes in 23 pairs, and you've got 18,000 genes. So they're not the same, but they seem to be linked in some way because they're following the same rules of inheritance. We now know that genes are physically located on chromosomes, and how we know that, I'm going to hit you with in more detail on Monday. But for right now, I'm actually going to wind this down 2 minutes early. That's all right. I'll try to keep you late sometime to make it up to you. I know you really want to get maximum value for your educational dollar, and I will stop the recording now.